ladies and gentlemen. Today we are going to start talking about Chapter 27, which is going to really cover the basics of our two domains, bacteria and archaea. Keep in mind when we classify organisms, we're talking about three major domains, your bacteria, your archaea, and your eukarya. So we are going to focus here today on bacteria and archaea, and we'll talk more about eukaryotes or eukarya later this year. So when we're talking about your domains bacteria and archaea, we're really talking strictly about prokaryotic cells here. So let's just kind of give us a refresher. So prokaryotes, right, they're unicellular organisms, so they are single-celled, free-living. Um, some of them do live in colonies, so they will congregate together and sort of divide and conquer certain activities of life if they choose to, but they're not required to live in those colonies. It just kind of helps them be a little more efficient overall when they do. And these guys thrive almost everywhere, from your skin to your guts to every known surface on Earth, man-made or otherwise, to talking about surviving in ice, to the depths of the oceans, to you name it. We have pretty much found bacteria everywhere. And in fact, when we're looking for life extraterrestrially, so exoplanets, other life outside of Earth, this is what we predominantly believe is probably out there. Because it just really seems to be able to live and survive everywhere here on Earth, so it would make sense that that would be something that could transfer elsewhere in our solar system or the universe for that matter. They do have a relatively wide range in terms of diameter. They are microscopic for sure, but when we look at their range and size, we're talking about 0.5 typically to about 5 micrometers, which is a pretty significant range for the type of organism we're talking about here. And if you think back to evolution, these guys were more than likely the first organisms on Earth. So how do we classify them? There's a handful of characteristics that we sort of progress through, particularly when we're talking about identifying a particular bacterial or archaeal species. Um, One of the most common ways that we look at prokaryotes is to throw them under a microscope and identify their shape. So some of the most common shapes of prokaryotes are listed here. So we can talk about them being spherical, or what they are classified as cocci or a coccus, and that's what you're seeing up here, right? If those come in pairs, they're diplo, and it can be diplococcus. It can also be these rod-shaped bacilli, which are these guys here, a bacilli or bacillus. So those guys can also be diplococcus, diplobacillus. Um, if they are found in clusters like this, so they sort of look like a bunch of grapes, then they're staphylo, is the prefix, and again, those could be staphylococcus or staphyloBacillus. Or they can come in these chains, which is where we get the prefix strepto. Again, or strept, right? And so that can be streptococcus. If you've ever had strep throat, that's what that looks like. They can also be streptobacillus. Third one are these spirals. All right. What they call sporilla, if they're more just a common comma-like shape, to the spirochetes. If you know anything about the bacteria that causes Lyme disease is caused by a spirochete, and it looks like a corkscrew, or a helix. If you think about like DNA, it's this big spiraling on itself. And so those are the major shapes. They're not the only ones. Some of them kind of look close to crossing one or the other. Um, but for the most part, they pretty much stay within these major categories. So that's what we look for first. The next thing that we're going to look at is their cell wall structure. So before I talk about how we use that cell wall structure as a classification scheme, let's just back up for a step, and just talk about what it is and what it does for the cell itself. So really, when we're looking at that cellular structure of a prokaryote, remember you've got your cytoplasm in the middle, you've got your cell membrane, and then they have a cell wall. They can have additional layers beyond that cell wall structure, and we'll talk about some of those potential options in just a moment. But they will have at least cell membrane, cell wall. Those are pretty standard. So they're there to really help the cell maintain shape. It provides protection, so it's an extra layer of not only just stability in terms of, think of osmotic pressure, right? So if they are constantly living in a watery environment, which is the case, then they need to be able to prevent themselves from bursting into a million pieces when they end up in those hypotonic environments, right? And do you remember what that term means? Well, let's back up. So hypotonic, remember, is when you have more water outside your cells than you do inside. And so as a result, we end up shoving additional water into your cells via that process of osmosis, and it causes the cells to swell up, right? There's more sugars and salts and ions and things dissolved inside the cell and less water as a result than there is outside. And osmosis is a passive transport system, and it's going to constantly be working to try and equalize out concentrations of solutes on either side of that cell membrane. Um, and so these cells had to evolve ways to survive in situations where they were getting too much water rushing into their cells. When it happens to us, we don't have that protection. We don't have that cell wall, sort of elastic band that keeps it from taking in too much. Plants do. So these bacterial cells have this cell wall, just like the plants do, to prevent themselves from bursting open. So it only allows them to take in so much water before the pressure builds up within the side of the cell and just prevents additional water from moving. That being said, it does not help on the reverse side. So when we go from a hypotonic being too much water outside and it's rushing in, isotonic, if you remember that term, and that's coming back in a couple of weeks. Isotonic, it's equal on either side. Hypertonic, right? Hypertonic is the opposite, where now I've got too much salts and sugars and things outside my cells that it's sucking the water out of my cytoplasm and into the environment instead. So while the cell wall will prevent the cell from bursting when too much water is going in, it can't work the opposite direction. And so if those cells lose enough water, they can actually get their cell membrane to shrink away or uh, detach from the cell wall, what we talk about as a plasmolysis process. Um, and that's going to inhibit reproduction. You're basically killing the cell, you're dehydrating it. Um, so when we talk about dehydrating foods, this is exactly what we're doing to the food, but what we didn't know back in the 17, 1800s, or go back even further, we've been doing this for millennia almost. Um, we use it as a way to preserve foods and salts, in sugars, in even just drying the foods in air. And we're causing these bacterial cells that would live on that food and ultimately cause it to spoil from being able to reproduce, because we're dehydrating the bacterial cells right along with the cells in the food itself. So it's a great way to preserve that food and prevent it from spoiling, because you're killing the bacteria in the process. Okay, so let's talk structure for a second. When we talk about the cell wall structure of bacterial cells, they contain peptidoglycan. And that peptidoglycan is going to be a really important way for us to start distinguishing certain species of bacteria. It's a type of polymer, specifically a combination of carbohydrates, so some modified sugars, and they're cross-linked with these short polypeptides, so short proteins. Again, this encloses the entire bacteria because it's the main component of the cell wall structure. It can also be as an additional anchor point in the cells that extends from its surface. It can also help anchor things internally. Keep in mind, peptidoglycan is pretty much a bacterial thing. Archaea, since we're talking about those also in this chapter, contain a polysaccharide and a protein that are similar, but they are not peptidoglycans. So it is a slightly different uh, sugar protein complex that exists in the cell walls of archaea. So we can use that then as a way to start distinguishing bacteria from archaea, is looking for whether or not the presence of peptidoglycan exists. So we use peptidoglycan as a classification mechanism not only between bacteria and archaea, but then also to start distinguishing the major species of bacteria itself. So if you had an unknown bacterial species that you cultured from somewhere, whether it's a medical patient and you're trying to find out what they're infected with, an environmental component, whatever the case might be, typically the first step is to take that sample, grow it on a petri dish, and then look at it under a microscope. And when you're looking at those first initial slides under a microscope, you more often than not will probably end up doing a gram stain. And that gram stain is going to be able to narrow down two major classes of bacteria. So gram staining is simply a method that's going to distinguish between the two major structural components that we see in cell walls in bacteria, and it has to do with how much peptidoglycan they have. So we talk about them as either being gram positive or gram negative based on the way that they handle the staining procedure, and it's going to come down to how much peptidoglycan is in their cell walls. So when we look at gram-positive bacteria, they look like this. Here's their plasma membrane, and then pretty much overarchingly, their cell wall is peptidoglycan and not much else. And so they will ultimately have a much less complex cell wall structure as a result of those huge layers of peptidoglycan. They also end up being a little less toxic. They're a little more susceptible to the vast majority of antibiotics that we have, and they will typically stain purple in this protocol, and we'll step through that protocol in just a second, and I'll explain why the gram-positive tends to trap that purple stain. Gram-negative, on the other hand, has a bit more complex structure for their cell walls. So here's their plasma membrane. They've got this thin layer of peptidoglycan between, and then they have another outer membrane of phospholipids and carbohydrates and lipopolysaccharides and so on and so forth. So they have a bit more of a complex structure to their cell wall. As a result, that less peptidoglycan is going to impact how much of this purple stain they're able to hold on to. And so they end up looking pink based on a counter stain that we use when we look at them under a microscope. And these guys tend to be a little bit more toxic and a little less susceptible to the vast majority of antibiotics that we have because they have this extra complex layer on top of the peptidoglycan. So this would be what you would typically see under a microscope after doing a gram stain procedure if you had a mixed sample. So you've got these gram-positive bacteria here that are purple, your gram-negatives are the pink guys. And so again, we're typically doing this as one of the first steps for identification of an unknown species because it gives me two pieces of information from one slide. It tells me something about its cell wall structure. So when we go through classification, one of the biggest things we look at for bacteria is their gram stain. Are they positive or negative? And then I also can start digging into another major component of their classification, and that's their shape. So I can kind of kill two birds with one stone, if you will, when we do this as our sort of first step for identifying an organism. So it's a really valuable tool. I say here in medicine, but to be real, it's a valuable tool across all microbiology. So if we're going to talk medicine, though, which is often I found for kids in the past, they're most interested in that aspect more often than not, then the gram-positives tend to be a little more susceptible, right? So not only to things like antibiotics, but also to your own body's defenses. So remember, those gram-positives have that less complex cell wall structure, and so that just makes them more accessible for targeting, both with medications as well as your own body's ability to get at them. The gram-negatives, on the other hand, having that additional lipopolysaccharide layer above their peptidoglycan kind of really blocks out a lot of the ability for the targets for the antibiotics, as well as your own body's defenses. So they have a little bit more protection on all sides of things. So let's talk about how they work. Let's talk about the basic steps through a gram stain procedure. So first and foremost, you would have to, what we call, fix that bacteria to a slide. Basically, we need to get it to adhere so it's not going to get washed away by these repetitive liquid stains we're going to put on them. So that's where they're talking about this fixation piece. So just getting it to stick to a slide. Basically, you warm it up enough that it sticks. Then we're going to treat it with this purple stain called crystal violet. And that crystal violet at this point will stick to both gram-positive and gram-negative. If I stopped the staining procedure here, they would all look purple under the microscope. I'm then going to treat it with iodine, and that iodine is going to help adhere that crystal violet to the peptidoglycan. So it's a chemical reaction component that's going to allow that crystal violet to bond more tightly and stick to the peptidoglycan layer. Again, I stop here, and everybody's going to look purple. We then do a decolorization stage with alcohol. And this stage you've got to be careful with. You only should be doing it for a very short stint. You want it to do just enough to sort of wash away the initial top layer of the stain. Because gram-positive has such a thick layer, if you do this correctly, you shouldn't be washing away all of their purple stain, but you will be washing away the top layer of purple in a gram-negative, and so they will appear colorless. If you do this for too long, though, you can get the purple to wash away from the gram-positive as well. So this is a very important step to be done correctly, otherwise you basically negate what you've done previously to that step. The final step then is to do what they call a counter stain, and stain them then with an additional stain, in this case called something called saffron, which is sort of a pinkish red. And what you end up doing is that counter stain then sticks again to all cells. So if you decolorize these guys enough to the point where that purple disappeared, these guys would appear pink. But if you've done this decolorization stage correctly, that pink is still there on these guys, but that purple is so much stronger of a stain in terms of its color that you'll basically see them as purple. But because these guys have lost that, they don't have enough peptidoglycan there to hold on to that gram or that crystal violet through that decolorization stage, they take on that saffron stain instead, and so that's where the pink comes from. So when you look at this, right, all of these pink guys are that gram-negative because they didn't have enough to hang on to that crystal violet. And so they took on the saffron stain, and that's all you see. The gram-positives, on the other hand, these purple little guys here, had so much peptidoglycan that you probably only washed away that top layer of purple. And so that purple is still predominantly strong enough to shine above and beyond that lighter pink, and so they end up looking purple under the microscope as a result. So it's a nice way to identify their cell wall structure relatively quickly. And again, we use that as one of the first lines of identification. And then we look at them under the microscope at the same time, looking at their shapes, and now we've got two major components of identification taken care of. Other things that we can look at as we're identifying these guys is to look for other unique structures that some of them have, others do not. One thing that some prokaryotes, some bacteria have outside of their cell wall is to have a capsule. And this is basically a sticky polysaccharide sugar complex that surrounds their cell walls. It's going to be an additional layer of protection and evasion if they are pathogenic to their host. And it allows them to just stick to surfaces as well. So not only does it provide them just some extra protection, but it's kind of like giving them a big layer of Velcro or a big layer of glue. And so it allows them to adhere to the surfaces of cells or just surfaces in general for that matter, so that they're not getting washed away. Other external structures that these guys may have, fimbriae, which are these just numerous appendages. All right, so they kind of look like these big hairs. Again, going to help them to attach to surfaces, sort of like little suction cups. These tend to be more numerous and shorter than these pili. And these pili can also act as a attachment component, but they will also serve as a way for cells to go through a process called conjugation, and we'll talk more about what that means later. So when they're using it for that conjugation process, you may also come across the term a sex pili or a sex pilus. It really is the closest way that bacteria can have quote unquote sex. And really, we're just talking about the ability to exchange DNA from one cell to another. And we'll go into some more detail on how that conjugation happens. But that pili is kind of like a straw being shoved into two cells, provides a channel to shuttle DNA from one cell to the next. We may also look for their ability to move. Do they have motility? And there's some testing that we can do to determine that, both inside certain types of culture media as well as looking at them under a microscope. And so do they have the ability to perform taxis? Remember, we talked about that in animal behavior back in ecology. So can they move to or away from a particular substance or stimulus? And a lot of times their motility is based on chemotaxis, so response to chemicals, moving towards nutrients or oxygen, or moving away from something that would be toxic to the way that they survive. About half of the bacterial species that we know of at this point are capable of directional movement. So that means that they would have the structural component called a flagellum. They may have multiple flagella, they may have a single one, they may have them all over their body, they may have them in opposite ends, maybe in one end. They can really have a huge diversity in terms of how many flagella they have and where they're located on their body. And so these are just three potential examples of how this could work. But they do allow for locomotion. They allow for their ability to move. And so when we look at the structure of a flagella, here's actually an electron microscopy image of this component right here. So it anchors itself into their cell membrane through their cell wall, and they basically use these as like corkscrews. They spin, kind of like a rotor on a boat, or a propeller on a boat, a motor. And so it spins in these circular corkscrew motions, and that's what allows them to move. When we look at this flagellal structure, we can see some evolutionary differences in the proteins that are made up of them. From archaea to bacteria as well as eukarya, they all use different proteins, and they are very significantly different. So we do believe that they all evolved independently of each other, which is kind of interesting. It wouldn't be what we would typically think of when we talk about progression of structures, but because they are so very much independent in the way that those proteins are structured, we think they actually all independently evolve, which is kind of cool. And then remember that these guys are prokaryotes, so they lack all of that compartmentalism that we see in eukaryotic cells. There is really not much to their internal structure. They don't have a true nucleus. They don't have organelles. Their genome is just floating around in their nucleoid region in the center. That being said, they are certain species that will have sort of these infolds of their cell membrane. So this sort of start to some internal structure, specifically acting either as respiratory or thylakoid or mitochondrial-esque membranes to allow them to do things with oxygen, with their energy consumption, energy production. So when we look at these guys, sort of pre true mitochondria, pre true thylakoids, in the sense that they exist in eukaryotic cells, but some of that internal organization looks very similar to those organelles. Again, giving us some support to that endosymbiotic theory that these guys would have been, those organelles would have been free living prokaryotes at one point, taken in by a larger host, and that symbiotic relationship that developed over